So we receive here around 2,000 samples per day. Okay. Over here, what we do is cytogenetics. We do also. We'll see. Okay. So this is a common reception area. Okay. And now you enter inside the lab. So as you see over here, you have now the microscope here. You have here the, let's say, the preparation of the sample. Okay, so we're talking about these kind of samples that just go inside this. They are cut in there, they are prepared, and then they go into the microscopy there and okay. So this is one of the biggest labs in Portugal, and this detects only pathology, right? And this only detects, this is the lab that detects the most cancers in Portugal, okay? So they are really professional with this, and then when you have these sports, they go to the doctor, they have a report, okay? And they are going to be looking at the samples, and they and now we're going to like the biology lab. Okay, so this was just a beginning. Okay. Now we are going inside the molecular biology lab. What they have over here is the samples, okay? The samples that come from all the drive-thrus, all those stuffs. Now they come in here. They are prepared in here. Okay? Over here the samples arrive. It's the early morning, so we don't have a lot of samples. Okay, some of the samples. Hola, Carlos.
Hello.
Hello.
So Carlos is the molecular biology director in here. Okay, so it's responsible for all of this lab and is going to accompany us and do all the questions for you. So what we have is this. So this is a COVID Copa swab. Okay, and afterwards it will go into the lab. Okay, something like this. So over here we receive this one is from, I don't know, I think it's from a drive-thru. Okay, and then they will go into preparation. So they will go into the preparation. Let's say the
Inactivation
inactivation is made in this step,
Yeah.
okay, inside with one.
when it's needed, because sometimes we use when we have swabs with like inat, they already have inactivation, and so in that case we only transfer to those tubes that are either inactivated or either just a transfer from the inactive.
So over here, what we use nowadays is the CG, which is a Korean company as well. So we are using mainly CG material. Okay, we have also these equipments from CG, this Hamilton one that comes from there. Okay, and what we do is we do the extraction in here automatically. Okay, so these equipment do the extraction, and they are going to do the amplification in the next row. Okay.
All of this is integrated. There is no data inferred manually. It's all connected with LIS and data read from the vials, and then send the data to the thermocycler that
Yep.
you see. Because it's a full solution.
This is the final part of the process. That equipment over there discharges the data in here, okay? And now there is the amplification, okay? of course, that there is a small plate. If you want to show, Carlos.
I can show you what we use.
Yeah.
Is an interface that reads the data generated from the equipment, and the interpretation is full automated.
Okay.
So nobody has to see and interpret it manually, and there is no manual introduction of anything. And we just select and do it HL7 export to the LIF. So there is nothing.
No people involved. It's just an automatic process that
Yeah.
goes from the extraction until the report of the case. Okay. So this is one of the labs. We have one more. Okay, that is on the other location. And this is it. This is very important for us. Okay. There is one machine also that I want you to see. Okay, the other one,
Okay.
which for us is very important. So this process takes 6 hours. Bom dia. Ela vai. So each sample 6 hours, 96 samples in one plate. Okay, this one 45 minutes. Each cartridge like this. Okay.
Maybe you already know because it's well known.
Yeah, it's well known. So each one of the swabs comes in here, okay, and you put in the machine. 45 minutes, you have the result. Okay, so these are express tests, and those ones are the common PCR tests. So usually we are interested in these two. Okay, for example, we have an island in Azores that wants to do 1,000 tests per day. The thing is, if we have this, it will be very simple to put inside and give the result in the airport. That process over there, as it has two machines, it's more specialized, it will be more difficult. Okay, so there are differences between these tests. We use the two. So this test is more expensive, that test is cheaper. Okay,
We
so
don't.
like this.
We do a lot of things with this platform already. When we have urgent case, even for chlamydia, HPV viral load, HIV, whatever, we use almost all the portfolio. So for large numbers, it's not good solution. For this long tail of small is very good, and everybody like it. Yeah, I know. I can share a comment of a huge competitor
Yeah.
of this. I can if because you are not you are not from Portugal, I can say one day Rose fed the only thing wrong with this is that it's not from them. It's the so it's a very nice robust system.
Yeah. So usually we are interested in these two types of systems. So a system like those, completely integrated from the extraction to the amplification to the analysis. Okay. If it has an algorithm to analyze, the better it is. Okay, like that one. And then we are interested in this one, which is really fast, really quick, and we can give the result in a very short time. Okay. So these equipment over here are around, this one will be around 150k, okay, with this position, with these 16 positions, okay, but there are equipment like this that can go almost to 300k, something like that, okay. So usually we are putting this inside the hospitals, inside, let's say, airports, things like that, so we give a very quick result.
And even if you want, we can arrange a box model like this, and you can pick 4 models, for example, and put it in another. You can split whatever you want.
Yeah, exactly. Okay. So clear. Let's go into the room and let's talk about the thing.
Thank you.